Okay, let's unpack this. For the last few years, the biggest headlines in the green economy have all been about synthetic biology, engineering life.
Hmm.
We've been doing this deep dive into sustainable materials, bio-based ingredients, and there's this pervasive assumption that the hardest part is the DNA.
That's exactly right. The focus is almost always on, let's say, biological elegance. You're designing the perfect organism or mapping the ideal plasmid.
Right.
The idea is you convince a microbe to make your molecule in a nice, clean lab flask.
Yeah.
And everyone just assumes if you solve the biology, the rest is just plumbing.
Exactly. You get this beautiful lab result, the yield looks fantastic in a 5-liter batch, and you think, great, we've cracked it, now let's build the plant.
And that's when it all falls apart.
So what does this mean for the startups trying to cross that infamous commercial valley of death?
Well, here's the core,
Hmm.
and it's kind of the surprising finding from the sources you shared. The overwhelming majority of these promising bio-based projects, they don't fail because of a glitch in the DNA. They fail because of problems where messy biomass meets real steel. The dominant risks aren't biological. They are mechanical, logistical, and thermodynamic.
They're in the field.
They're in the field, they're in the truck, and they're in that massive reactor vessel.
That is a fundamental, almost terrifying shift in focus. We're moving the conversation from the lab bench to the factory floor.
We have to.
So our mission today is to give you, the listener, a shortcut to understanding those physical, those non-biological barriers.
Precisely. We need to unpack 3 critical engineering challenges that,
Right.
time and again, just sink projects. First, designing with a multi-product biorefinery mindset.
Okay, one.
Second, managing the operational chaos we call feedstock entropy. And third, surviving the unforgiving physics of industrial scale-up.
And if you can master those 3,
If you master these 3 pillars, you dramatically de-risk your whole business.
let's start with that first one then. The biorefinery mindset. When I think of a big chemical plant, I think of it making one thing, one high-value substance. So why is that hero molecule approach so flawed in the bio world?
It's because the input material, whether it's agricultural residue like, say, sugarcane bagasse or forestry waste, it's inherently complex.
Okay.
And it's cheap per ton, but really expensive to move. Unlike crude oil, biomass is this complex matrix of cellulose, hemicellulose, lignin, water.
It's not uniform at all.
Not at all. So a business model based on pulling out just one high-value product and then discarding the rest as waste. It just instantly makes your economics incredibly fragile.
So if I'm an investor, why should I care if the plant monetizes that residual stream? Isn't the profit in the main product?
The profit might be in the main product, but the resilience is in the residuals. And what's fascinating here is that a modern biorefinery has to be conceptually similar to a traditional petroleum refinery. Its core economic goal must be to turn every fraction of that feedstock into something useful. You have to maximize the revenue per ton you bring in, not just per molecule of your flagship product.
Can you walk us through those fractions if we take, say, some kind of plant material, lignocellulosic material? What are the key revenue streams there?
Well, you typically see 3 main economic streams. First, you have the highly structured fibers, which are mostly cellulose and nanocellulose.
And what are those used for?
These are premium. They're used for high-value applications, like specialty barrier films and packaging, advanced composites, that kind of thing.
Okay, so that's the structure. What's next?
Second, you get the sugars. These are released from the cellulose and hemicellulose after you pre-treat it. And these are the main feed for your core fermentation.
That's where the hero molecule lives.
That's often where your hero molecule lives, yes. It's producing everything from organic acids to advanced biopolymers or even high-value proteins.
And the third fraction, the stuff that sounds like waste. The lignin, the residuals.
That's the most critical stream for stabilizing the entire business model. Lignin is the rigid, sort of glue-like component of the plant. You can use these residual solids for internal energy generation.
So you burn it.
You burn it to provide the necessary heat and steam to power the entire facility. This reduces your reliance on natural gas, and it stabilizes your operating costs. You basically turn an expense into a cost-saving stream.
So the lignin isn't just waste, it's risk mitigation, it's operational independence.
Absolutely.
Let's imagine a facility then. It's getting wheat straw. So one stream goes to nanocellulose, one goes into the fermentation tanks for bioplastics.
Hmm.
And that third stream, the lignin-rich solids, provides the heat and energy for the first two. So the whole business model really rests on that slate working together.
It does. And think about it. If your single flagship product hits a market downturn, the entire plant doesn't necessarily have to shut down. The diversified slate, the nanocellulose, the bioplastics, the internal energy, it provides margin and resilience. And that is exactly what investors are looking for.
And the sources really emphasize co-location. Why is building the biorefinery right next to a farm or a processing hub such a make-or-break decision?
The simple answer is density. Biomass is bulky and light. Moving straw bales or sugarcane stalks over long distances is just incredibly costly.
So you want to be close.
If you can co-locate your biorefinery right next to, say, a sugar mill, you get that biomass already aggregated. You slash your trucking costs. Plus, you stabilize the feedstock quality because it's coming from a predictable, known source.
Which helps with traceability.
Exactly. It simplifies the traceability you need for those premium sustainable products.
That makes perfect sense. Okay, let's shift gears from logistics to our second big engineering headache, what you called feedstock entropy. What exactly do you mean by entropy here?
When a chemical engineer talks about entropy, we're talking about disorder, or in this case, variability. Agricultural residues are chaotic inputs. They're not like refined chemicals.
You're messy.
It's very messy. Feedstock entropy is all about the massive variability in these agri-food byproducts, and that variability introduces crushing operational risk.
And what variables are we talking about here? It's more than just being a little wet, right?
Oh, far more. We're talking huge swings in moisture content depending on the harvest season, the weather, how long the bales sat out in a field.
Okay.
We're also talking about the intrinsic chemical composition, the ratio of cellulose to hemicellulose to lignin, which can vary by region or even the specific cultivar. And crucially, you see high variance in mineral content, ash, dirt, and literal contaminants.
Like rocks.
Rocks, stones, residual pesticides, yes.
So if you're running a plant that processes straw, every single truckload that arrives is a fundamentally different chemical input.
It is. I mean, think of it like trying to bake the exact same cake every single day. But every time you open the bag of flour, the flour has changed its moisture content, its protein level, and its contamination.
And that would change everything.
It changes how severely you need to pre-treat the material. It alters the final yield of sugars you can extract, and it drastically impacts fouling and the whole energy balance of the plant.
That sounds like a technical annoyance, but you mentioned it has monumental economic pain points. You highlighted that process modeling shows that this variability can cause, what, 20 to 40% swings in the minimum selling price?
That's a terrifying number.
That's huge.
And a number that keeps CFOs up at night.
Yeah.
The MSP, or minimum selling price, is what you have to charge to break even and satisfy your investors. If your model shows that just under normal operational variability, not a catastrophe, just normal day-to-day changes, your MSP can swing by 40%. You're unbankable.
A 40% volatility means you could be competitive with fossil fuels one day and completely irrelevant the next. No investor is going to fund that.
That's the barrier. Temporal and spatial variability in biomass quality is now recognized as a major barrier to attracting serious investment unless you explicitly manage it. You can't just design for the perfect average input anymore.
Okay, so if the variability is unavoidable because, weather happens, what are the strategies to denoise the feedstock before it hits the sensitive parts of the process?
Well, first is platform diversification. You have to qualify multiple feedstock suppliers across different regions or different harvest seasons.
To build a buffer.
To buffer against local crop failures or bad weather. Second, ruthless quality control at the gate. You have to define tight incoming specs, moisture, ash, particle size, and you have to be ready to reject or discount loads that don't meet them.
You have to be willing to send the trash back.
You do. And the third strategy is using the plant itself as a buffer. You use pretreatment, blending, and substantial intermediate storage to literally smooth out the variability over time.
So you're building a shock absorber into the front of the plant.
Exactly, so that the core bioprocess sees a homogenized, predictable stream, regardless of the chaos happening outside the gates.
That is a powerful concept. The whole design philosophy shifts from process what comes in to homogenize it first, then process it.
You have to design for the extremes, not the mean. Feedstock is a probability distribution, not a single number. If you design your facility for the average bale, the worst 10% of bales, the wettest, the dirtiest, will inevitably blow up your yields, accelerate fouling, and just destroy your economics.
Okay, we've covered the external supply chain. Now let's move inside the facility for the third big hurdle, the unforgiving physics of scale-up. What really changes when a process that works perfectly in a 5-liter lab flask gets moved into a 50 cubic meter industrial tank?
The environment the cells experience, it just changes drastically. The physics simply do not scale in a linear way. In the lab, you have perfect control. In the factory, you're fighting entropy, friction, and fluid dynamics.
So, challenge one. This is critical for most bioprocesses. Oxygen transfer.
Oxygen transfer, yeah. It's measured by the volumetric mass transfer coefficient, or ALA. In the lab, you can agitate like crazy, pump in tons of air, you get high kLa values.
Yeah.
But at industrial scale, tanks of 50, 100 cubic meters,
That's huge.
the power you can apply per volume is constrained by mechanical limits and just the sheer cost of energy. You can only spin the impeller so fast.
Right, before you tear the whole thing apart.
Precisely. So the collet typically drops, and it drops significantly. For highly aerobic organisms, the oxygen supply becomes the new rate-limiting step, completely overriding your beautiful genetics.
So the cells are basically starving for air.
In a massive deep tank, yes, it dramatically caps your productivity.
And then there's mixing. A lab reactor mixes in seconds. What happens in the large tanks that creates the second challenge? These spatial gradients.
Well, a large production tank can take tens of seconds, sometimes a minute or more, for complete mixing.
Yeah.
And that lag time creates massive spatial gradients.
Meaning it's not the same everywhere in the tank.
Not at all. The concentration of everything, substrate, product, dissolved oxygen, even pH is not uniform.
So if you're adding, say, concentrated acid to control the pH.
Correct. The cells near the inlet get blasted with a pH shock before the entire volume can neutralize. The cells are traveling through these high-stress neighborhoods.
Good neighborhoods and bad neighborhoods.
Exactly. They might go from a rich, substrate overloaded zone to a zone of complete starvation and then get hit by a pH swing. All of that causes cell stress, triggers unwanted side reactions, and leads to variable product quality.
I love that analogy. What are the other physical limits besides oxygen and mixing?
Two more big ones, both related to geometry. First, heat removal. Fermentations are exothermic. They generate heat.
Okay.
As you scale up, your surface-to-volume ratio plummets. In a tiny flask, heat dissipates easily. In a massive tank, you have very little relative surface area to remove that heat. You risk overheating the whole batch, which can kill the process instantly.
You've effectively designed a gigantic thermos.
You have. And the final issue is fouling and flow. Non-ideal flow patterns lead to dead zones, areas where liquid barely moves. This affects conversion, but more importantly, these dead zones dramatically increase the risk of your products sticking to the walls and heat exchangers.
Which means more downtime and more cleaning.
More downtime, more cleaning costs. Exactly.
So the real job of the scale-up engineer is to fight all these natural physical constraints, the lack of surface area, the difficulty of mixing, to try and recreate that perfect lab environment.
That is the core job. You are fighting physics to create what you could call cellular democracy. Your goal is to make sure every single cell gets a fair shot at the food and oxygen, and that no cell gets stuck in a stress zone.
So if the physics are so unforgiving, how do teams mitigate these risks before spending hundreds of millions? They're not just doing trial and error at full scale, surely.
Absolutely not. The emerging solution is a massive increase in the use of digital twins and CFD-led design. That's computational fluid dynamics.
So advanced simulation.
Very advanced simulation. These tools combine complex fluid flow models with your biological models to simulate the mixing, the oxygen transfer, the temperature fields, all inside the massive tank before you cut any steel.
Which means you can iterate your mechanical design, the impeller shape, baffle placement, all in a virtual world.
It saves millions years of development time, and it prevents catastrophic failures. It lets you tune your mechanical design against the known physics rather than just fighting blindly after the concrete is poured.
What a fantastic deep dive. So we've established the bottlenecks are overwhelmingly engineering and supply chain, not biological. We've talked about embracing biorefinery thinking, quantifying and managing feedstock entropy, and mastering scale-up physics.
And if we connect this all back to you, the listener, the founder, or technical lead, the final challenge is one of mindset. You have to treat worst-case feedstock and worst-case hydrodynamics, the worst possible mixing, as fundamental design inputs.
Not as exceptions.
Not as annoying exceptions you hope you can avoid.
Why is that difference in perspective so critical?
Because at commercial scale, those edge cases become your Tuesday. They become your routine. The only way to build a robust, bankable plant is to design it to handle those low probability, high impact events reliably without crashing your yield.
So here's the final provocative question for you to explore as you move forward. Have you actually run models and experiments for the wettest biomass you might receive, for the lowest possible cellulose content, for the slowest, most inefficient mixing regime in the largest tank you plan to use? The teams that really embrace this thinking, that quantify that entropy and that master scale-up physics early, those are the ones that turn nice lab curves into robust, bankable commercial plants.